Hey everybody, this is Raquel Flaskus. I'm coming to you guys for another video in our DNA video series for the Chesapeake DNA Study. So thank you so much for tuning in. So we have just sent out a study update email that should have come across your inbox earlier this week. I'm so excited because we're planning our meeting to come down to the St. Mary's Historical Society and give a presentation on research updates for you guys. So part and parcel of planning out this event, we've been thinking a lot about what are the types of things that we should be talking about, and fundamental with all of this that I think we're going to be revisiting is what are our study hypotheses. And it's really important to know these things because we want you to understand the things that we're looking for and why it's important. So we're going to be talking about this during the event, but I thought maybe it would be good to give kind of like a pre-shadow before our event coming up at the end of February. So our first study hypothesis is that what is the genetic ancestry of persons with 17th century Chesapeake ancestry or heritage? So because we know if you were eligible for the study, you were able to trace your genealogy back to the 17th century Chesapeake, Maryland, Virginia, or Delaware, either through documentary evidence or strong oral histories. Now what we want to do is, because you've been able to trace these lines back to this time period, what does your genetic ancestry look like? Are we seeing predominantly English ancestry, Irish, West African, Central African, or really is the subset of a person's ancestry much smaller that has Chesapeake origin? So are we seeing a predominantly different ancestral makeup than what we would expect to see with somebody with a British European descent or an African descent from Central or South, or Central West Africa? So we want to kind of put together the genealogy with the genetics. So you want to see, where do they align? Where do they not align? Now, how we're going to also do this is compare specific markers in the genome that are known to be associated with colonial populations. So that being said, what we're going to be doing is looking at a model off of paper that was done in, I think, 2015. It's called the Fine Scale Structure of British Populations, which basically controlled for ancestry, like what we're doing. So they only worked with persons with ancestry to the 1800s. And they were able to see and to show that there is fine scale, so very, very, very tiny amount of genetic clustering for persons that have been able to trace their ancestry all the way back to the 1800s and still live there today. So those populations, for instance, in Wales, look slightly different than those, let's say, in the Isle of Skye or in the south of England. Now, that being said, it's very small, but it's still apparent. So what we're going to be doing is seeing if we can kind of look at those markers that they looked at and see how they overlap with populations living in the Chesapeake. And that's for an English subset. We're going to try to also do this with other colonial target populations, so France, also involving countries in West Africa and South Africa and Central Africa and South Africa as well, to kind of see, you know, what is this genetic makeup of persons and how can we really parse out what this kind of Chesapeake ancestry genetically looks like. So that's kind of first. This is our basic survey question, is what does the genetic makeup of persons who control for this ancestry genealogically look like? Okay. Our second one is then, how does this compare with genetic diversity of actual 17th century and 18th century archaeological populations? Now, this study question is kind of an adjacent study to what we're doing here. So my dissertation has multiple parts, as dissertations always do. We did work with a lot of archaeological populations that are being curated by the Smithsonian Institution, the Maryland Historic Trust, and also the Calvert County local government. Now, these are archaeological persons and burials that have already been excavated. Professionally, the remains are being curated under the protection of different institutions that I have mentioned. I've applied for permits to do DNA testing on all these individuals. We have in total about 40 persons from the Chesapeake, and we have another 36 from Charleston, South Carolina, if anyone is interested. And so we're going to be comparing the genomes of those individuals with our study group here in St. Mary's, just to see if there's any overlaps. We're not trying to specifically identify specific descendants, but we just want to see as a group, how has our genetic diversity changed over 200 years? Do we see that it's largely the same, or do we see that there's probably a little bit of difference just because it has been about 250 years? We have a lot of other genetic makeup that's coming in during that time. But it's going to be this kind of temporal comparison of how do genes change in a colonization context. So when we have massive amounts of European individuals and then also African individuals largely forcibly transported, how does that change the genetic makeup of a certain time and space? Okay, so that's one, which is an exciting one. So there are multiple little studies within that where we do case studies. So in a previous video, I talked about a Forbes article that came out that summarized our work in Avery's Rest, which is a 17th century archaeological site in Delaware, near Lewes, Delaware, kind of near Rehoboth Beach, Bethany Beach area, if you guys are familiar. And so that one we looked at European and African descended relationships through genomes, through mitochondrial DNA, and then also interpreted the osteology and the archaeology of the site. So we're going to be doing small subsections of that, but larger we're going to be comparing totally how do these archaeological persons compare with our study group. And kind of a second part of these study hypotheses has to do with kinship and relationships, right? So we're talking about ancestry, where are people coming from, what is our genetic background, right? And now our second part has to do with kinship, relationships, biological kinship, okay? And that's because persons, when they're related to each other, pass on a certain portion of their DNA. So we can test and look and see how much of these DNA overlaps are there, and thus how genetically related people are. So in our study, like we said before, we controlled for genetic ancestry, right? So we said, or we controlled for genealogical ancestry, excuse me. So we said if you had ancestry to the 17th century Chesapeake, you were allowed to be in this study. Now that we've controlled for that, basically we have a subset of the population living in this area that we know has roots, very, very deep roots to this area. And we can kind of remove all the other people that are more recent migrations, okay? Now, when we just have this core of persons that we know have been able to trace their ancestry to this specific area, how do they relate to each other? Now, that's important because only a handful of studies have looked at this, and what they found, similar to that fine scale study in England found, is that people are more likely to be related to each other when they have kind of deeper this more likely to be related to each other when they have this kind of deeper genealogical heritage, or they've been living in the same place for a long period of time. Just the nature of the beast. So people, if you're living in the same area, you have higher chances of being related to them just because of time and how families go. And we've all seen genealogical trees and how complex and interwoven they may be. Now, why is this really important? Because it helps us understand the nature of how microevolutionary changes in genetic diversity are shaped over time. How does kinship and kind of this intermarriage or we call this admixture in genetics, so kind of switching of genes between two people, how does that shift genetic diversity over time, right? So we have a population that we can control for ancestry, which you guys are, and a population that is archaeological. We can look and see how they've changed. So what is different? What is the same? How are people more likely to be related in our contemporary population just because we've controlled for this ancestry? And so this in part and parcel will help us understand where people come from genetically, how people are related to each other, and then how does genetic diversity change over time in a over 200 to 250 year time frame. So those are our kind of main study hypotheses. We'll go over them in more detail during our presentation. But maybe next week I'll be going into our methods a little bit more if you guys are interested in kind of learning about how we actually answer those questions more specifically. But we'll also be sending we'll be videotaping the conference and the presentation for you guys for those who are not able to attend. So looking forward to seeing you guys all there. Hope you guys enjoyed this.